Welcome to the webinar. Today's USP 797, wait, what? That's interesting. A functional approach to quality sterile compounding is presented by Dr. George Smith, System Sterile Compounding Quality Specialist with Prisma Health Department of Pharmacy. Dr. Smith is a board-certified sterile compounding safety advocate. In addition to his position at Prisma Health, he is also the principal of Echelon Compounding, LLC, a consulting firm dedicated to advancing sterile compounding and technology. Dr. Smith's career has traversed many areas of pharmacy, including retail pharmacy management, pharmacy legal regulatory inspections, home infusion partnership, and multiple clinical leadership roles within the health system pharmacy. Dr. Smith has dedicated the better part of his career to promoting safe practices for medication delivery and raising awareness of the increased risk involved in sterile medication compounding processes. Today's webinar is accredited through ACPE and is worth 1 contact hour. CE credit is applied following completion of an evaluation link found on the last slide in the presentation handout. You can find the handout on the lower portion of the GoToWebinar toolbox under Handouts. We are recording this session and will include the recording and evaluation link in a follow-up email. Questions may be submitted throughout the webinar using the GoToWebinar toolbox on the right side of your screen under the Questions tab. We will answer the questions submitted following the presentation. Thank you for joining today's session.
Thank you, Amy, and thank you, ARL Biopharma, for your support of these wonderful educational sessions for our profession. It's really important that we continue to strive in this manner moving forward. So I've got some good news and some bad news. The good news is we're going to keep this to an hour. The bad news is really I have about 11 more topics that I'd really love to talk about in this section, but we're going to hit some of the high points here. Additionally, get out your smartphones. We're going to do some polling and do some interactive discussions with some questions. So get those out and be ready. When we get to that spot, I will instruct you on what to do. So let's get through the legal mumbo jumbo. I have nothing to disclose. As Amy said, this is worth 1 CE contact hour. And there will be some more instructions at the end regarding how to claim your 1 hour of CE. So our learning objectives for today, at the completion of this activity, you will be able to describe the history of the revised USP 797 chapter, discuss the purpose and enforceability of USP 797, recognize the elements required for compounding under the per approved labeling section of USP 797, section 1.4, and explain some various chapter elements that operationalize compounding for compliance and patient safety. So let's get started right now with a poll. This is going to get us warmed up. So you can join by the web using that website on the left. You can join by text. If you join by text, you are able to just submit your answer via A, B, or C, 1, 2, or 3 in the text once you receive the text that you've joined, or you can scan that QR code. And that will kind of actually follow along on your smartphone as to what question is up when I get to those questions. So far, my organization was 100% compliant with the 2008 USP 797 on the day before the new 2023 version to go official. And we're seeing some answers coming here and there. I think the role here is really looking at it from the standpoint of, we're at 62% false, 63. Looking at it from this is a journey, and there'll be a lot more to discuss as we get closer to that. But it is a long time coming that we've gotten to this point, and there'll be more to come as we go through all these years. So we're getting to almost 50-50. which is a little bit higher than I would have thought, but that's outstanding because as this became official on November 1st, 2022 and November 1st of 2023, on November 1st of 2022, we knew what the new compliance features would be going for 2023. So moving on, let's move on to our very first question. What is USP 797? Is it a set of rules and regulations regarding sterile compounding enforceable by regulatory agencies? Is it a set of rules and regulations of sterile compounding contained in Chapter 21 of the Code of Federal Regulations, Sections 210 and 211? Is it a chapter that describes the minimum standards of sterile compounding adopted and enforced by all states' boards of pharmacy and other regulatory agencies? Or is it a chapter that describes the minimum standards of that can be adopted and enforced by state boards of pharmacy and other regulatory agencies? Seeing that right now we're at almost 90% of number 4, with a little bit of 3 and a little bit of number 2. So great, let's keep moving, and we'll talk about these answers as we get to the next session. So USP sterile compounding standards describes the minimum standards to be followed for the preparation of compounded sterile preparations for both human and animal drugs. The definition is really important here because it is defined as combining, admixing, diluting, pooling, reconstituting. It does include repackaging or otherwise altering a drug product or bulk substance to create a sterile preparation. So it does include repackaging, USP standards. These apply to all healthcare personnel in all healthcare settings. USP only sets standards that are then incorporated into regulations. Essentially, there is no USP enforcement team or surveyors or inspectors that work for USP. These are standards that can be incorporated into state boards, regulatory agencies, accrediting agencies to, inspect and survey on this process. So number 4 was the correct answer. Number 1, a set of rules and regulations. It is not. It is not rules and regulations, they're standards. I know we've been taught, and it is the truth, that anything less than Chapter 1000, or I guess 999, is enforceable, but it is not in and of itself enforceable by USP. Number 2, which was took 0 on the poll, is basically chapters defining 503B outsourcing facilities or compounding pharmacies. 3 was a little tricky, putting in there that it is adopted and enforced by all state boards of pharmacy. That's where I think the rubber is meeting the road here with a lot of different in places. For example, in South Carolina, we have our own sterile compounding part of the Pharmacy Practice Act. We have not adopted as of yet USP 797 as a whole. There are other states that have adopted USP, and so when it changes, that just kind of rolls in. And there are other states that are coming up with either more stringent or more released or lax rules regarding sterile compounding. So number 4, being that it can be adopted and enforced by the state boards of pharmacy, is the correct answer. Great. So this journey to where we've gotten here in USP, this is, looking at, we 13,000 stakeholder comments. Think about that for a second, that on these revisions, and For example, comments were, sent into USP sometimes by organizations. So maybe there was 10 comments sent in by from an organization, but not necessarily 25 people sending in the same comment. So a large stakeholder involvement. We went through 3 compounding expert committee cycles of 5 years apiece. There were 3 drafts of USP 797. It took 13 years to get where we are today with the official 2023 version of USP. 2 appeals, not that anybody's proud of that, and 1 remanding back to the compounding expert committee regarding beyond use dating. But what was interesting is what started out as USP 797 that had radiopharmaceuticals, it had non-hazardous compounding, is now 3 separate standards with USP 800 for hazardous compounding and USP 825 of the compounding of radiopharmaceuticals. So the journey has been long, but very, very worth it. And we still have a long way to go because this has just really started. I was asked in my role as the system sterile compounding quality specialist, what did I foresee the changes in my bandwidth a few months after November 1st in the role that I was in? Well, some of you that know me know that I'm an endurance athlete and do some crazy, silly stuff and do races. So I kind of thought about this from the standpoint that, November 1st of 2023 was the start line. Walking up to the start line was what really November 1st was all about. Everything up until that point was getting out of my car, getting my stuff ready, and walking to the start line, maybe even doing a little warm up. But really, the journey has just begun. We've got a long way to go. A quick little review here, and I know what you're saying. George, haven't we beat this to death? Well, I guess I do believe that the hallmark of adult education is repetition. And these categories and how they're broken up and described and distinguished in USP is the foundation of the chapter in how we're mitigating both the risk and increasing safety. So wanted to say that right off the bat that category 3 is out of scope of this presentation, and that we'll really be just discussing category 1 and category 2. So if we look at it from the standpoint that category 1 is the least controlled environmental conditions, and as we increase categories, we're increasing our stability assurance levels, and we're able to assign longer beyond use dates, which are important to operationalize what we're trying to do in any manner in which we are compounding. The beyond use dates for category 1, 12 hours, no more than 12 hours at controlled room temperature, no more than 24 hours in the refrigerator. There is no frozen allotment for category 1 CSPs. Category 2 gets a little bit more complex. We throw in terminally sterilized CSPs, but for the most part, not doing any type of terminal sterilization and or sterility studies. We're looking at 4 days room temperature, 10 days in the refrigerator, and 45 days frozen. Now, do a lot of different things with this, terminally sterilize, sterility testing. That gives us a lot more that we can look into and gives us a lot more flexibility. So let's jump into our, first little thing of kind of talking about, some of these things, I'm going to be honest, are a little controversial, and I hope you get that out of this, is that some of what we're talking about can be done in a lot of different ways. So we've been taught for a lot of years, looking at USP, that facility design is the key. And it is. And it does dictate really how we should look at our CSP categories, but it doesn't necessarily have to be. Let's say you're a community hospital. Let's say you're a vet practice or a long-term care pharmacy. Got a lot of different ways of looking at this. I think the question that needs to be asked first off is, do I need Category 2 BUDs? And I think that's where looking at your operation, looking at your patient population, and deciding, if my operation doesn't require Category 2 BUDs and I have a clean room suite, do I necessarily have to assign Category 2 BUDs by keeping my clean room suite to the requirements and the specifications in USP 797? And I don't think so. And what I mean by that is we can look at this from a very practical perspective. And here's a few elements of comparison. A lot of things are the same. Training and competencies for Category 1 versus Category 2, personal hygiene and garbing are the same. It really boils down to the environment. So let's say you do have a clean room suite and you have decided that I don't necessarily need to do Category 2 compounding. I do not need those dates. Is there some possible either justification or thought process that could go into analyzing what this could look like? Well, if you look at certification and recertification in Category 1 as an SCA, you only have to certify and recertify the PEC. In Category 2, all classified areas, your anteroom, your buffer room, have to be certified and recertified as well. From a microbial, biological, air and surface sampling perspective, in an SCA, it's only the PEC. Surface sampling in the PEC and frequently touched areas in your SCA is best practice, but that is a good way to determine your environmental load and what your flora is. But in Category 2, we're looking at much more robust sampling. The anteroom, your buffer room, your PECs, and all of that's based on, specific locations based on your square footage or cubic meters, based in an ISO standard, and of course the frequently touched surfaces. So you can see this becomes a much larger process for you having Category 2 compounding. Cleaning, disinfecting, and sporicidal application, again, there are some similarities, but there are some differences. Daily floor cleaning in the SCA, depending on where your line of demarcation is, could be a very small area. If you have even in a large clean room suite that you designate as an SCA, all classified areas into compound Category 2 have to be cleaned daily on the floors. Walls, doors, shelvings, monthly in all classified areas, Category 2. Shelving in all of that other than the ceilings not being soiled in Category 1. So there is a difference there when you're looking at what it's going to take to prepare Category 2 compounded sterile products versus Category 1. And there is a thought process here that maybe you don't have to do this if you're not necessary for your patients. Beyond use dating is really the key here. That's where you start. For a Category 1, 12 hours and 24 hours and no frozen, is that something that your organization or your pharmacy can do? Is that going to lead to increased waste? Is that going to lead to, expiry of products on your floors or in your pharmacy, that's an analysis that you'll have to do. But that doesn't necessarily, it may be something worthy of doing if you look at what the next, the impact of maybe converting all of that. So what are those possible justifications? Well, I broke them down into two areas. I think they're both financial, and I believe there are processes involved in both of those, and some of them overlap. For example, decreased room certification and recertification costs. If you look at doing a clean room suite with the full gamut of things, depending on your certifier, it's $4,000 or $5,000 for the room, all the HEPA filters, everything that it entails with leak testing, air changes. When you look at surface sampling, there'd be less surface sampling if you are not a clean room suite. Microbiology costs, sending it to your own lab, sending it to a third-party lab. Those services do cost money. Cleaning supplies cost. You're having less supplies to use. You're using less disinfectants, less sporicides, less disposables and consumables in a smaller area. And resources. I'd love to have a poll here, but I didn't. But who has resource challenges? I think we all do. I think we're having trouble finding resources. I think we're having trouble keeping resources. We have great people inside of our pharmacy, and a lot of times all they want to do is make products to take care of patients. And I think those are some of the tasks that are involved with documentation and sampling, and all those ideas may not bring a whole lot of feeling of self-worth or mean worthwhile work to someone who's doing some of that stuff. Processes, decreased cleaning time, decreased documentation. And the old adage of if it's not documented, it doesn't happen. So there's a renewed vigor in USP on documentation and specific things that we must document. Less standard operating procedures, less things to change, less things to look through, less things that have issues. And a possible decreased risk of patient harm due to mitigating viable particle contamination and not having these products sit around for longer for that microbial life cycle that, when microbes are contaminating into a bag or a syringe, and the opportunity for them to proliferate and then be a problem to a patient and be pathogenic. So those are some considerations. I've known of a number of my colleagues that are greatly considering this, just from the standpoint of, this may make their organization and their life a little bit easier inside their pharmacy. Their hair's on fire for a lot of different processes going on, and USP and sterile compounding is just one of those processes that needs our attention. And if we can make it simpler, then that is also a good thing. So question number 2 is, according to USP 797, and there's a lot in there about conventionally manufactured products and how to deal with them. Here is an actual package insert or prescribing information from a vial of erythrocin for use. And so the question is according to USP 797, this vial can be stored up to 14 days in the refrigerator. True or false? So here's our poll. And this is exactly what I would have expected. This is another one of those controversial subjects that we're going to discuss. Great, so we're really about 90%, give or take a couple percentage points here, think that that is false, that this vial cannot be stored for 14 days in the refrigerator. Let's talk about that. So while this, example is a vial, let's pretend that this same thing it could be used also for a bag. So here's our per approved labeling in section 1.4. Here's the information that we have in the prescribing information. Let's look at this from a practical perspective. What does section 1.4 say? So preparation per approved labeling is compounding does not include mixing, reconstituting, or other such acts that are, and here's the important part, performed in accordance with the directions contained in approved labeling, package insert, prescribing information, or supplemental materials provided by the product's manufacturer. So preparing a conventionally manufactured sterile product in accordance with the directions in the manufacturer's approved labeling is out of scope of this chapter only if the product is prepared as a single dose for an individual patient, and the approved labeling includes information for the diluent, the resultant strength, and the container closure system and storage time. That's what the chapter says. That is verbatim what the chapter says. So what does this mean? How do we interpret this? Well, let's start with some current compounding definitions. So Food and Drug Administration, in essence, is mostly concerned about creating new drugs. So drug compounding is regarded by their definition as process of combining, mixing, or altering ingredients to create a medication tailored. To them, that is more like a new drug for the needs of an individual patient. Missing from that definition is repackaging, by the way. USP defines compounding as combining, admixing, diluting, pooling, reconstituting, repackaging, or otherwise altering a drug product or a bulk drug substance to create a sterile preparation. Two similar yet different definitions of what compounding really is. It really would be great if we had a standard, but alas, we don't. Sorry to disappoint you guys. So let's go with what is not meant by USP's preparation of per approved labeling. What is not meant is to restrict a pharmacist's professional judgment regarding assessing the sterile preparation, physical stability, chemical stability, therapeutic stability, toxicological stability, microbiological stability, and beyond use dates. And you're thinking, whoa, George, you just need to stop. We have a couple things here that I think we can talk about. We have seen this kind of morph over the last 10 years as to a really misunderstanding and a leap for all intents and purposes for some things. So, much has been talked about and much has been said, much has been surveyed and much has been inspected under the guise that if the prescribing information or the manufacturer, the package insert has a BUD in it, then we must use that BUD. And we've beat that a lot, and we've talked about that a lot. That's not what USP is after, nor is what this is about either. As an aside, has anybody looked at a norepinephrine package insert lately? We use the heck out of that in critical care in the hospital. It talks about making it as 4 milligrams in 1,000 milliliters. Okay, I'm used to now 16 milligrams in 250 milliliters. So, let's package inserts aside. There's a lot to be said for that. The second part of that is back in 2015, CMS and the Joint Commission started surveying some weird stuff, and it really revolved around the succinylcholine vial, in which we had a lot of data to show that while the manufacturer said that the succinylcholine vial was only stable or only good, however we wanted to call it, outside the refrigerator for 14 days, we had copious scientific evidence. I had actually done a stability indicating study on succinylcholine using automation, repackaging into smaller syringes, in which it was very stable, greater than 94% at 90 days. But the Joint Commission, CMS came in and said, we're not doing that. That's where I think we got a lot of confusion. We got a lot of controversy, and we started thinking, and we have pharmacists calling me now in my specific role saying, hey, I just saw this package insert for epinephrine that says epinephrine in 250 mils of diluted, in normal saline and stable for 24 hours. Well, we know that there are plenty of good stability indicating studies out there for something like epinephrine, that there is stability more than that. So going back to this section, which kind of went down a little rabbit hole there, what is not meant is really trying to restrict us from our professional judgment. Now, I guess the question becomes well, if that's the case, then why is it in here? What happened? Well, without mentioning any names, hint, hint, the agency that is responsible for drug approval wanted to make sure that their, air quote, approval process carried as much weight as possible. Really, they were looking for the agency is mostly concerned about creating and approving new drugs, but they wanted to make sure that we understood that if a manufacturer had gone through all the proper tests and all the proper processes, much like a USP monograph, then that was not considered compounding and did not have to take place or fall under USP 797. Now, extremely confusing. What does that really mean when we're looking at this erythromycin? And what is the practicality of that in the situation? I love checklists. And so here's my checklist for compounding per approved labeling under section 1.4, which is what we're talking about here. It must contain, so the per approved labeling requirements are, it's a single dose for an individual patient. We have a diluent listed, specific diluent, a resultant strength, a container closure system, and a specified storage time. So reconstituting this 500 milligram vial with 10 mils of sterile water, is that compounding, or is that approved per approved labeling, and that we can go by what the prescribing information says? So let's quickly look back at our prescribing information. And what do we have? We have basically a single dose for an individual patient, maybe. That's up to you. That's up to you if you've made it. If you're trying to say, I want a 250 milligram dose, well, that's not a single dose. We can't do anything with that rest of that vial. But if we're going to reconstitute that entire vial and give that vial in a bag, compound with it to a single patient, then that is okay. But that is up to you. Is the diluent listed? Well, yes, it's 10 mils of sterile water. Do we have a resultant strength? We do. 50 milligrams of erythromycin activity, or basically stating that 10 mils into the powder is going to give us 50 milligrams per mil. What about the container closure system? Well, we're talking about a vial here. We're reconstituting a vial. And we do have some specific storage time. So that storage time is 2 weeks, 14 days in the refrigerator. So the question becomes, can this vial be stored up to 14 days in the refrigerator once reconstituted by these directions in the package insert? And the answer to that question is true. And I'm just as confused about some of that as you are in a lot of cases. While the answer to that may be true, it begs the question, should you do it that way? So those are some things, again, another checklist. I like to look at this from the standpoint of, what's important here. So before 1938, package inserts did not really go through the same scrutiny and vigor that they do now with creating the information that we have in the new drug approval process. So not all approved labeling or package inserts are created equally. So something to consider. You're saying, George, USP 797 is about sterility insurance, not just stability. So, but what are you talking about here? Well, these manufacturers in today's world are required to do some type of, USP 51 microbial effectiveness testing for anything that's either an antibiotic or has a preservative in there, and they must pass those tests. So that is a process to look at as well. Was this done for erythromycin back in when this package insert was written? I don't know. Don't have any idea. Another question to ask. Are new things typically done with USP 51 and passing those tests? Absolutely. The third part of that is not all stability studies are created equally. We've learned a lot over the years of what those elements are. Looking at stability indicating studies, we look at force degradation studies to isolate some of the excipients or the impurities. We would like to know what those are as physical and chemical breakdown occurs, not just a potency over time. So not all stability studies are created equally. And then finally, asking yourself, why? Why do I want to do this way? Do I need to do it this way? Best practice would be if you get all the way down here and say, hey, maybe I want to do this, is call the company, see what their process was for getting stability studies done. Did they do USP 51 microbiological effective testing? When was their package insert approved? I think the why becomes important here. Would I do this routinely? Probably not. I just don't think it's useful. Erythromycin goes on massive shortage. Not a bad idea. I probably would, especially if I have a lot of patients that are on erythromycin. Maybe I would do this like this. Saving money as an option, I think that's a little risky. I think we're kind of taking out of context the safety and what we're trying to accomplish. But again, this is what per approved labeling is about. It was, one of the things that I scratch my head about and shake my head and wonder, was it worth it to be in there? But I guess that agency's liaisons decided they wanted to have this included in USP. So a lot more to come on this over the years and time to come. A lot of people are talking about it and what it is, but just remember, it's not meant to restrict us in any way from the daily things that we do using our own judgment. So let's move on to question number 3. Which statements are correct regarding proprietary bag and vial systems? So think mini bag plus, think vial to bag, think advantage. So docking and activation of proprietary bag and vial systems in accordance with the manufacturer's labeling for immediate administration to an individual patient is not considered compounding. So which statements are true? Docking for future activation must be performed in an ISO 5 environment contained in an ISO 7 buffer room to be allowed to assign the BUD based on the manufacturer labeling. Docking for future activation is considered compounding, and docking for future activation performed in a PEC contained in a SCA are to be assigned only a 12-hour room BUD in accordance with Category 1 compounding. So looking at some of the answers. Okay, so this one's a really tricky one as well. And we've had a lot of different people talking about this, and really, I think you can take it a couple different ways. And let's kind of talk about this from the standpoint of what is required for this. Proprietary bag and vial systems, docking and activation of proprietary bag and vial systems in accordance with the manufacturer labeling, immediate administration to an individual patient is not considered compounding. Proprietary bag and vial systems for future activation may be performed in an ISO 5 PEC. Beyond use dates for future activation cannot be more than specified in manufacturer's labeling as long as they're docked in an ISO 5 PEC. And an ISO 7 environment is not required to give dating that is up to the manufacturer's recommendation as long as it is done in a PEC. Let me say that again. An ISO 7 environment is not required to give the dates up to the manufacturer's requirements according to USP 797. So do you have a PEC? If you do, that's all you need. This has been a little confusing. A lot of people have been talking about this, and we've taken the term that we can do this in the PEC. This is afforded in USP 797. So 1 and 3 is actually the answer because docking for future activation, according to the manufacturer, only requires a PEC. It does not require a ISO 7 environment or a Category 2 environment. Very confusing. A lot of different people are talking about this in a lot of different ways. Again, if you want to be conservative, but according to the chapter, it can be done up to the manufacturer's dating just in a PEC. Couple more things, moving to thermometer and temperature verification. This one is one that baffles me a little bit. There's some verbiage in here. All temperature and humidity monitoring devices must be verified for accuracy at least every 12 months are required by the manufacturer. And what are those? Locations where components are handled and or stored. So we're thinking of ambient locations, shelves, any type of refrigerators, including med rooms, automated dispensing cabinets that have refrigerators on them, things like that. Freezers, of course. Locations where CSPs are handled or stored, same as above. And then ambient locations where compounding occurs. So your clean room suite and your sterile compounding area. That monitoring equipment should be verified for accuracy. Or must be, excuse me, must be. Should to me sometimes means, yeah, you got to do that. But according to the chapter, we have shoulds and musts. Shoulds are recommendation, musts are got to do it. But what is verification for accuracy? Well, I tried to look at this from a scientific perspective. And the verification ensures the process or equipment operates according to its stated specifications. So think about a thermometer now. Calibration ensures that the measurement accuracy of the instrument meets a known standard, okay? And it really boils down to is what I believe USP intention here was to make sure that the monitoring devices for the temperature and humidity are verified as accurate against a known standard. So they really do intend for some up to point of we're calibrating this equipment without going out and saying it. So continuing on, looking at from a pharmacy temperature monitoring automation perspective, those are sophisticated systems and can be calibrated and validated every 12 months. Couple examples are TempTrack, Thermo Fisher Scientific units. I had Reese Scientific at Prisma for a number of years. We had those calibrated and validated every year, every single one of our temperature and humidity devices. Interestingly, though, only section 9.3.4, component handling and storage, includes the phrase calibrated. The rest of the areas where it talks about temperature, it doesn't. It only talks about the word verify. So what do we do? How do we accomplish what we're trying to do here? So what are our options? Obviously, a big proponent of temperature monitoring automation, which is, the ideal solution. They're pricey. Not everybody can have these things, depending on the situation. But the system is calibrated and can be calibrated and validated every 12 months. And you typically get a nice little binder of all the documentation, all the delta changes, any revisions that are made, and everything that goes along with that. So nice, wrapped up in a nice, big, pretty bow. But what if you don't have that? What are your options from here? Well, example, a standalone thermometer with, recommending a minimum and maximum temperature and recording that so you know what had happened if there was any excursions and what that looked like. So your options really are, you can replace that every year. So that example right there that I'm showing is a $80 to $100 to $120 wherever you can find it. Solution. You can replace that every year. It does come NIST certified, but can't be calibrated. You could purchase, specific thermometer or temperature monitoring and humidity equipment that is calibratable. That means you're basically sending that back in every year or based on at least every year to get it calibrated and receiving it back. Now that does obviously cost you a little bit of money to do that in time that you're going to have to send it in and get it back. Another option is to purchase, investing in a source thermometer or source hygrometer that is calibratable every year, and you could use that to verify the accuracy of each one of your standalone thermometers. That is also, those are a little bit more expensive. They're hundreds and hundreds of dollars for each one of those. You're going to have an SOP in place and defining your process and lots of documentation, but that's also an option. Things that are not said in USP, but things that are practical for, what this really does look like. Regardless of whatever you choose, in the essence that you are going to have an SOP that outlines not only the plan, but the documentation of this verification, and it needs to be retrievable. So those are some of your options for, how do I measure temperature and humidity in my spaces, and how do I verify for accuracy? Our final question of the day, as we're getting close to wrapping up, is moving on to differential pressure monitoring and/or monitoring devices. So according to USP, differential pressure monitoring and/or monitoring devices must be verified for accuracy every 12 months or as required by the manufacturer, cannot be measured with analog gauges. Digital devices are required, are not required to produce quantitative result either electronically or on display, should provide a pressure gradient of at least 0.02 inches of water column from the SCA to the ambient space, or none of the above. See these numbers coming in. We're at 75, 76% must be verified every year. Right, so we're about 75% with none of the above, coming in at 13, and should provide the pressure gradient of 0.02 from the SCA to the ambient space. So here, a couple examples of differential pressure monitoring. You've probably seen something of this manner in your pharmacy or in other pharmacies, measuring differential pressure. So what does, this is what I see. There is something missing from that digital display that's really important in USP. And what is that? That is, we must have a number. So I have gotten with our engineers at Prisma, and we have that Pressura digital display on the right, and I am asking to make sure that that display does show me some quantitative results in looking at, so I can trend, so I can see it, and what that really, really means in looking at it. So differential pressure monitoring and verification. Interestingly, the chapter is silent on the verification of accuracy and/or the calibration of these differential pressure devices. Silent, nothing to be said about it. Analog gauges are allowed. I guess the drawback to that would be, yeah, I get to see a number, but does not alert me if in the middle of the night and I'm not 24 hours, or even if I am, if my HVAC system goes down. And there's lots of issues with that. Could be out of range. Quantitative results are required to be recorded and documented, whether that's a continuous recording or not. I think it's important to understand that from a practical perspective, maintaining and looking at the differential pressures in clean room suites are the only way of really knowing that your HVAC system is operating properly. Yeah, it may get a little bit warm. You may feel the humidity go up if something's going on, but that is a very good quantitative measure as to what's going on with the HVAC system. I actually believe and suggest that this is actually more important than monitoring my temperature and humidity inside my clean room or inside my SCA. Yeah, I'm all for comfort. I'm all for making sure that, you know, temperatures are below 68 degrees and below 60% relative humidity for comfort, and to make sure we're, you know, doing our best not to proliferate organisms by sweating and shedding particles and things like that. But I think it's important to understand that this is one of those environmental facility designs that makes a big, big difference. And there is no requirement for a pressure gradient for an SCA, except for the containment SCA, so the hazardous drug room, going from ambient space. And that one, for the CSCA, that requirement would be that range of negative 0.01 to negative 0.03 inches of water column to make sure that we have negative pressure in that room. So answer to question number 4 is actually none of the above. So nothing said about being verified, can measure with analog gauges, are required to produce some type of quantitative number that's good to trend. It's good to see what's going on. As your HEPA filters become more efficient, they actually get clogged, and that, you know, space in that room, the CFMs may be less, and you'll notice that in sometimes declining differential pressures. And we do not have to provide a pressure gradient for an SCA to the ambient space. So in summary, USP is currently official, and it's many years in the making. I think they've done a great job with a lot of elements, and I think it's a fantastic revision that we've moved into. USP 795, 797, 800, and 825 are enforceable by legal or regulatory bodies, but not in and of itself a rule, regulation, or law. USP's per approved labeling, as you saw, is met with confusion at best and misunderstanding completely at worst. And just remember, it's not intended to limit pharmacy practice or pharmacist judgment. I, along with a lot of people, and I hope you are, very appreciative of the number of places in the chapter where it's our professional judgment is allowed, whether it's in our SOPs on who a checking pharmacist is versus a compounder versus a you know, supervising pharmacist. I think those are some great things with those freedoms. And, you know, granted, I'm a type A pharmacist and would love to have had that in black and white, but I am appreciative that, you know, if you've seen one hospital pharmacy, you've seen exactly one hospital pharmacy. We're all different, a lot of different practices out there. And I really do think that USP is a great starting point for a minimum standard. But I challenge myself, I challenge my organization, and I challenge you to understand that we need to strive for best practice, that, you know, these are just some of the standards we need to hit. But in everything we do every day, it really is about our patients and what we're trying to provide them in a quality, sterile, compounded product. So from there, I'm going to see if we've got any questions. Question number 1, we got a few minutes. Do you think that per approved labeling section and the additional BUD of 4 hours for immediate use compounding will allow pharmacy to move much more compounding outside of pharmacy? That is a concern. From a personal perspective, I've thought of that as a concern. I've done a number of years of work in Europe, and a lot of their compounding is done outside the pharmacy. But I can tell you from the Joint Commission's perspective that they will be looking at that pharmacy is very, very involved in the compounding process, and that we as a profession are to not just start moving stuff to our nurses and to other locations inside our facilities to do real routine compounding. So that is a great one. Only got a couple minutes. Does the differential pressure need to be manually documented, and how often, if so? As long as it is recorded continuously and is readily retrievable, it does not necessarily have to be documented. However, in my organization, I am asking our managers and our compounding personnel to look every day, whether that would be logging into a system or looking and documenting and making sure that our HVAC system's working as it's supposed to. But it is once a day on days that compounding occurs that we should be at least continuously recording it or documenting. I'd go to every shift if you're running 24 hours. A lot of best practices in there, as we know what this can do. And and secondly, what is your downtime procedure if you find a problem, and what does that look like? So we're right at the top of the hour, and I want to thank you for your time. I hope everybody has a fantastic holiday season. I will take these questions and answer them, and hopefully I can get, this will be part of the process being sent back out to you for the questions, but I thank you for your engagement, and I hope you have a fantastic day. Turn it back over to Amy.
Thank you, Dr. Smith, for this fantastic session. As a reminder, CE credit is applied following completion of an evaluation. A recording and evaluation link will be sent 1 hour after today's session. Thank you, everyone, for participating today.